Good afternoon. We're solving questions taken from old sessions from 2016, 2017. But you'll find when you solve paper two, you'll find that questions keep on repeating. The same question repeats like 4, 5, 6 times sometimes, right? So in biology, solving paper two is very, very rewarding because you may just get almost all the questions in the exam, already seen them before. So, first question. They gave me stem and root, and they wanted me to do what? They wanted me to identify the stem and the root in each case. So, let's just sketch a root first. It looks like this. It has an X sign in it, like this. And that X, ironically, is xylem. You see those circles I'm drawing? These are phloem. In the stem, it would look like this. And you'll have the xylem and phloem in what we call them vascular bundles, where the inside of the vascular bundle, the one I'm just shading here, xylem, and the one to the exterior is phloem. So that's xylem, that's phloem, that's a stem. Okay, that's a root. If some leaf comes in the exam, then it's also easy. Phloem is always near the floor in the leaf. Xylem is at the top. So take a close look at the diagram and tell me which one is the right one. Which one is the correct labeling of xylem and phloem? What do you guys think? You need to figure out that the shaded part is going to be xylem, right? The shaded part, because they said the water is being colored. They took a stem like this and they put it in color. The color will run only in xylem. So which one has that shaded part correctly labeled as xylem? Absolutely. Okay. All good? Second question taken from kidney. Now we got three labels here, X, Y, and Z. X is a blood vessel that delivers blood to the kidney. You know what that blood vessel is called? That's called renal artery. The one that takes the blood away from the kidney is renal vein. This is not a blood vessel, this is the ureter, okay? Now, let's just make a quick comparison between artery and vein, renal artery, renal vein. In terms of volume, the renal artery would always have a greater volume in terms of blood, I mean here. Why? Because when the blood arrives in the artery, it would take some water, some salt, some urea. Some of those will be removed from the blood. So obviously what would return back here in the vein is going to be less. What about the ureter? Ureter will have the least volume. Do you know why it's going to have the least volume in the ureter? Because the ureter only has some water, some urea, some salt, right? That's going to be the least amount of volume, okay? So which one is the correct option then? Is greater than passing in X. That's wrong, right? I'm comparing Y here, the vein. It should be, think about it, should be this one, right? Do you get it? It says that slightly less than that of X. So the vein is less than artery in terms of volume and is way more than the ureter. The ureter will only have leftover, some water, some urea, some salt. Is this good so far? Now, let's first, for this question, it's about the kidney still. Let's just identify those vessels. I got a blood vessel going to the liver. If the blood vessel is going to the liver, that must be what? That must be renal. Sorry, what am I saying? Hepatic artery. It's the liver. That's the renal artery, okay? The blood vessel that takes the blood away from the vein, away from the liver, I should say, hepatic vein. And the kidney is going to be renal vein, okay? Now they're asking me which vessels contain blood with the highest and lowest urea. That's an important question. How do you tell where would you have the least urea and where would you have the highest urea? Think about it. Urea will be least after it has been removed. Okay? Think about it. Which blood vessel would have the least urea among all those four? This one, right? That's because urea has been removed from the kidney. So that will be, now I'm thinking about the highest concentration of urea. Where will I expect to have the highest of concentration? It should be the one where urea has just been made. Do you know where urea is made? Do you know where urea is made? Urea is made here in the liver. So I am expecting it to be the highest after it's been made. So this, the lowest, this one is the highest. For that reason, I am gonna go for option B, okay? So the highest is 1, the lowest is 4. 4 is the renal vein. Why did I pick the highest as 1? Because that's where the urea is made initially. And why is the lowest is 4? Because that's where urea has cleaned, removed from your blood. Okay? We're gonna do a couple of inheritance questions. Usually those are the ones that take longer. And I said that before, if you're doing the exam, keep that question about inheritance, keep it the last thing, because it takes some extra time. Here it is said there is codominance. What does it mean when I say codominance? Codominance means both alleles, this and this, are equally dominant. And this one here is the recessive. Do you remember blood groups? We used to have A, B, equally dominant, and O is recessive. Okay? A, B are equally dominant. They want me to figure out how many different genotypes I can have and how many possible phenotypes I can have. Let's first consider the genotypes. I could have AEA, I could have AFAF, okay? And I could have a combination of E and F. That's the codominant. Now let's use G here. I can have a combination of EG, FG, and GG. These are all possible combinations you can have between those three alleles. So how many is the total? 2, 3, 4, 5, 6. How about phenotypes? How many phenotypes? Are there 4 or are they 6 phenotypes I can have? This is one possibility. This is another. E and F is the third because they're codominant, meaning they're going to produce a third phenotype. Remember, AB, blood group A and blood group B, they produce someone's blood group is AB. That's a third phenotype. EG is going to be E, Why? Because E is dominant to G, right? FG, F is dominant to G. But this last one is a possible phenotype. So total number of phenotypes I have is just 4. Again, why did I consider EG as a phenotype? Because it's going to be the same as E. If someone's blood group is AO, his blood group is A. Someone's blood group is BO, his blood group is B. See? Okay? Check your earlobe. If it happens that you have attached earlobe, that's recessive. And if it happens to have like a free earlobe, mine is attached, I believe, yeah. If it happens to be a free earlobe, that's dominant. Okay? So we have a family tree, like the one I have here, and I want to figure out how many of those are heterozygous. How many members of the family are heterozygous? Now, how do you figure out? Do you remember eye color? Eye color is like the easiest to understand. With eye color, if parents have brown and they end up with a child with blue eyes, then the parents must be heterozygous, okay? This applies everything. This applies everywhere. If parents have one eye color, the children have a different eye color or any feature, I almost dropped it, then the parents must be heterozygous. So let's apply this rule here. Let's apply it onto the diagram and tell me how many people are hetero. So what I have here is these two, right? 1 and 2. 1 and 2 have a different child, see? They're both free and they have attached. In addition, 7 must be hetero, right? Because his child all these children are actually attached earlobe. So he must be B capital, B small. Because if he was B capital, B capital, every single child would have been just like himself, just like herself. They're all free earlobes. All right? So again, parents of children that have different trait, they're always heterozygous. And if some parent was married to recessive, if he was B capital, B capital, why keep saying it's a she, 7 is a she. If she was B capital, B capital, and her husband 6 was B small, he has to be B small, then every single one of them would have been free earlobes, but that's not the case. Be hetero. Okay? Someone's telling me that she has seen people with one attached, one free. I don't think this exists. It's either both attached or both free. I don't think it exists. Different eye colors, yes, but earlobes, no. What is the chance that a child will have same blood group as his parents? So we have somebody's AO, somebody's BO, and same blood group as his parents. So let's cross AO and BO, like what I have here. AO, BO, so AB, and BO, AO, OO. So look at the Punnett square. How many of those have the same blood group as their parents? It is this and this. 2 out of 4. 2 out of 4 is like what? 2 out of 4 is like 50%, or 1 out of 2. It's the same thing, right? Okay. In some flowers, you have white and you have red and you end up with pink. That's also called dominance. Okay. So they got here, if a red and white flowers are crossed, how many will there be pink? You have red, you have white. How many you expect to be pink from this cross? It's like everyone, right? Every one of them will be pink. For that reason, that's like 100%. All are pink. Okay? Let's check 8. And I want you, once again, if you've forgotten, I want you to remember the series or the sequence of hormones in the menstrual cycle. We used to have first estrogen, like this, then, sorry, LH, I forgot it. LH first, then estrogen, then here it is, FSH, LH, estrogen, and the last one to peak. What was the last one to peak? The last one was progesterone, right? So they said that this lady, um, they want us to show where is their progesterone, where we'd expect her progesterone to peak. She had the menstruation here. Menstruation lasts like 5 days. She finished menstruation here. And you need to count 14 days from the start. 14 and 5 is like here. So you'd expect LH to peak right here, okay? Then where would progesterone peak? Progesterone will peak right after this. So I am expecting it to be anywhere here, right? Anywhere after 20. What about 27? No, that will be too late. Her cycle ends somewhere here, so you can't go beyond that. Okay? The increase has to be right after ovulation. So again, menstruation here, ovulation, remember that 2 weeks period, ovulation has to be 2 weeks, 14 days after menstruation. And following that, that progesterone will peak. This question here at the bottom is one of the worst answered questions. Most people don't get this one right. So take a moment and try to figure out what is the correct option. They gave you neuron P, neuron Q, okay? And they wanted you just to figure out which is the neuron and which is the correct direction. Now, just quick reminder, what's the correct order of neurons? It's always sensory, relay, motor. Always, always in this order. As sensory, relay, then the motor is the last one in this series. For that reason, you can't be going motor relay. That's wrong. Agreed? You can't be going motor sensory. Still wrong. P to Q, mo to C. It has to be that correct. But many people will go and choose C because C looks, whoa, nice. It's relay motor. No, it's not right. Because for some reason, they said QP. They said Q to P. So I'm going that way, which is still wrong. The only correct option is this, the last one, because they say Q to P, okay? It's been formatted not in a very friendly way, and they keep repeating it for some reason. They have very limited bank of questions, and they just choose them over and over again. Okay? This one should be not as challenging. Which substance is entirely absorbed? Meaning, which substance is not allowed to be in urine? That's what they meant. Which substance cannot end up in the urine? So, what am I going to pick? That's pretty simple, right? What am I supposed to pick? I'm going to pick this one. Okay? Someone is asking, she says, so most of the questions in the exam will, yeah, absolutely. It's mostly repeated questions. And it's a really good chance. You take the chance, you get 37, 38, 39 out of 40, it gives you boost. You score lower, it's just gonna drop you. You don't want that. Like me, when I'm making a test of multiple choice questions, I take from different papers and then I realize that the questions are the same. Question 11, they have like roots and shoots, and they said, how have the seedling responded? How are these seedlings responded? First one, I have done this in the dark, so they can't be responding to light, right? So the first one is gravitropism. They're responding to gravity. The second one, light, and they did respond. They went towards the light. So what is the responses like? They did respond to light, they did respond to gravity. I would go for both. Okay? And do you see how I read the question? I go to the direct line, the line that tells you what you need to do. If you just read this, check the diagrams, and then they read the line, is a waste of time. Go to the direct line, see what they want, and then check the details. Like this one. Which pair is controlled experiment? What's a controlled experiment? Meaning you have actual experiment with independent and dependent variable, and you have a one for comparison. So I need one set experiment and one set comparison, and I want to prove that light, they said phototropism, light is necessary. So again, my independent is light. So I need one for comparison with dark. I need light and dark as a set. Which one do I go for? Now, this is light. This is light. This is dark. This is dark. Which one do I go for? Do I go for set like 1, 2, or to the set 3, 4? If I picked 3 and 4, then I will face a problem. You know what the problem I'll face? Those shoots, you see the ones that put sideways? How will these respond? Those are put sideways, they'll go up despite being in the dark. It's negative gravitropism causing them. Look at the ones here. It's gravitropism make them going up. I can't be choosing this one because it's just going to confuse me. I wouldn't know. Is it light or is it gravity that's causing them to go up? Okay? That's why the only correct one is this one. Do you get it? This will cause that shoot to go up, not because only of light, it's because of light and because it goes against gravity. So that's not going to be a good choice to prove that light is necessary. There is a misconception about auxins. People think that auxins cause, what do you call it, cell division. And that's like a mistake. You can't, auxins have nothing to do with mitosis or cell division. They only cause cell elongation, okay? So what do we have here? We took a plant, we put it sideways, and when I put it sideways, the auxins will go down just because of the effect of gravity, like this. And then it starts to go against gravity, just like this. So automatically, I am nothing to do here with cell division. It's elongation, but where would you expect elongation to happen more? Is it going to be the bottom side or is it going to be the top side? It's always the bottom side. Why? Because it's has more auxins. Okay, so I'm gonna go for Q. Actually, D. Okay, more elongation in Q, the bottom side. Isn't that the bottom side where I have more auxins? Like here, bottom, you have more auxins. That will grow faster, right? Good. Now, again, one controlled experiment. And I think you got something like this in paper 4. What is a suitable control for this experiment? We are to study the effect of gravity, okay? This is the original experiment. This is my original experiment where you have dark and this you'd expect it to go against gravity. Do you see it? It's pretty small. See it? It's growing against gravity upward like this. I want a controlled experiment that proves that gravity is necessary. Straight away, get rid of anything that has the word light. Why? Because if there is light, then I'm no longer studying gravity. Once again, in an experiment, when I'm studying gravity, I turn off the lights and vice versa. So my case here, it must be dark and it must be rotating. Why should it be rotating? Because when it rotates, you will cancel gravity. See what I have here? As you rotate this one here, this means you cancel gravity. The plant is confused. It doesn't know where the gravity is coming from. It goes from all directions. Good? What causes the change between point S and T? That's a plant, seedling, and it's germinating, and we have a change in mass. But do you see what they've said? They said dry mass. Whoa, so what does that mean? This means it's the solid mass, has nothing to do with water. What causes dry mass to increase? So these are the seedlings. What would initially cause their mass to decrease and then to increase? From the start of this lesson, till the moment, it's been half an hour, I've lost mass. How did I lose mass? You're right now, you're not eating, you're drinking, you're losing mass. How do you lose mass all the time? You lose the mass because you respire. That heat that comes out from you makes you lose mass. So drop in mass is respiration, okay? What would cause an increase in your mass? When you eat, plants don't eat. How do they increase their dry mass? By making their own food, right? I can't be choosing transpiration, osmosis, because those are water. And the question is clearly saying dry mass. Dry mass means mass minus water, okay? Okay? Question 16 usually comes as the first question in the paper, and you don't want to get this one wrong. It's about the Mrs. Gren. Let's vote for this question. See how many of you get it right. Let's do a vote. Okay. Everyone picked either A and B, and two people for some reason picked C and D, right? Majority picked the right option, and it's option B. Why is it B? Why the correct answer is B? It's growth, right? It's pretty obvious there's growth, increase in size. And it's sensitivity. Why sensitivity? Because the roots went downwards and the shoot went upwards. Okay? No excretion. Do you see any wastes, gases, water vapor? Nothing about excretion here, okay? Aiden, Habibi, can you put this away? All right, thank you. Next, 17. What is the magnification of the photograph? That should be easy question. Magnification is what? They're not going to give you the formula like paper 6. It's just going to be memorized, which is super easy. So magnification is the image over the actual. Image 45, actual 25. 45 divided by 25, that is C. Okay? When you do this question, you really have to check the units. The units must match. Okay, just keep in mind. Very commonly, more commonly actually, is that they give you units that are not matching, like what I have here. So what am I supposed to do? Before I start, I match the unit 40 mm. If I'm going from millimeter to micrometer, I'll multiply by 1,000. It's going to be 40,000 here, okay? Now, 40,000 divided by 40, that's what? That's going to be 1,000. Okay, so check the units. Most of the times, they're going to give you units are not matching. So that's your task. Okay? Let's try one more. Here it says, diagram shows a high power. Diagram is images 80. And the actual is 160. But again, they're not matching. Straight away, what do? You multiply that 80 by 1,000. It becomes 80,000 over 160. That is what? That is, say, okay? $20. What is the magnification of this diagram? I got 2,000. They got 50 millimeters here, and I want to find the magnification. So 2,000 is the actual. When they say diagram, obviously that's the image. That's 50 millimeter to micrometer, 50,000, right? So 50,000 divided by 2,000. Okay? This question, for some reason, again, it's those two actually. Really, 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 just search them. You'll find them in 5, 6, 7 exam papers. And the one at the bottom. These are liver cells. And ironically, they're asking you to count cell walls in liver cells. So what do you must say here? They ask you for cell wall in a liver cell. Tell them, well, none, right? Same question. Look what they say. How many cell membranes you would have at this part X? Same picture as it is. They ask you at X, how many cell membranes do you have? We're going to say this time, we're going to say 2. Okay? Why there are 2? Because there's cell membrane of cell A, cell membrane of cell B. But the question here is just like asking about cell wall. The key thing. Use the key to identify the insect. Step 1, what do? I say wings are present. Do you see wings? Yes, 2 pairs of wings, right? I have wings. So wings are present. I'm going to go to 2. 2 pairs of wings, yes, they are. 1 pair, 2 pairs. I'm going to go to 3. Here where things go wrong. Are these circles? Do you see circles in their wings? Is this a circle for you? By any mean? No, right? It's round, but it's not a circle. For that reason, what should I pick? I say without. So most people pick C. I don't know why, but these are not circles. Okay? Again, this and this, very, very repetitive. Then comes this. Which one is partially permeable? And I tell you, like 50% of kids pick, for some reason, B. It's not B. Why it's not B? Because, first of all, that's plant cell, right? There's a cell wall. There is cytoplasm inside. And there is cell membrane. You tell me, what is B? B is nothing. B is empty space here. Because this cell has lost water. It's flaccid, it's plasmolized. The membrane drifted from the cell wall. So the cell wall, cell membrane, I should say, is C. And you know that partially permeable applies to cell membranes. Okay? "24. What's this about? This is about cholera. And they said, what effect does on water potential it will have when the toxin, remember that cholera, toxin causes the loss of chloride ions. So let's imagine that's the bacteria, release the toxin. This causes what? This causes the chloride, the salt, to come out. So all the salt is coming out. What happens to this area? That's the intestine here, right? The intestine is right here. What's going to happen as a consequence? Water potential here is dropped. Water potential in the blood is higher. Why again? The blood lost its salt. The salt went out of the blood. So water potential is high in the blood, very low in the intestine. So once again, what's the effect this one? This will say that water potential in the blood is increased, in the small intestine has decreased. Soon after, what's going to happen? Soon after, the water will move from the blood to the small intestine. Okay? So just reminder about osmosis. Water will always flow from where to where? It will flow from more water, which happens to be what? Higher water potential, to lower water potential. Or in other words, if you want to flip it, it's from an area of less salt to the area of more salt. Both ways work. So here's a quick question. It's about a cell, guard cell, and it said guard cells accumulate ions straight away. If someone is talking about movement of ions, what's the first thing should occur to you? Movement of ions. Ion movement is never diffusion, right? Movement of water, in what direction would you expect the water to move? They said the cell has accumulated ions. The ions moved into the cell. As a consequence, water will follow the ions. The area inside becoming salty, the water will follow the salt. Okay? What do I call a cell where it's packed with water like this? What am I going to call this cell? I say that the cell is becoming turgid. Okay? Okay? Turgid means it's packed with water. Turgid means it has gained water inside. Opposite of flaccid. Okay, 26. You can tell me this one right. I would appreciate it. Can we try this, please? Quickly, maybe? Faster than last time? So they wanted the highest water potential, okay? So where would you expect to have most water? Think about it that way. In which direction you expect the water to move in this diagram? Water will always move from soil, root hair cells, cortex cells, until it's xylem, right? And you know that water moves from higher water potential to lower water potential, right? For that reason, the area with the highest water potential is going to be here outside in the soil. And second highest, the root hair cells. Least highest is xylem. So again, you have to think of it that way. The highest water potential is the origin, the top, where the water originally comes from. The plant gets its water from the soil. For that reason, it's going to go soil, root hair cell, cortex, until it gets to the xylem. Same, this question, you'll see it's very, very commonly asked. And they wanted you to compare before to after. So this is the picture of the potatoes before, and you are to find how these potatoes look after once been immersed in water. Once again, first thing, think about how will the water move? Where do I have more water? Obviously, I'll have more water in the water. So water is going to move potato in, then to the salt. Okay? What's going to happen to the level of water in the tank here, the blue level? You would expect it to decrease, right? Because that's where the water came from. What's going to happen to the level of water here in the pink salty area? The level is going to rise. So in one area is going to drop, in one area is going to rise. So it's C. Okay? This question should take you less than 20 seconds. It looks complicated, long, but you could save a lot of time. How? If you could just take a look at the last slide. Which piece of potato, you have 4 pieces of potatoes, different concentrations. Which one of them has been put in distilled water? If you've soaked potatoes in distilled water, would you expect their mass to increase, decrease, stay constant? Obviously, it's going to increase, right? And when they put in distilled water has the highest of water potential, right? So what's going to happen? You would have the maximum increase, right? So A is distilled. Second highest is going to be 0.2. Next time they ask me about 0.4, it's here. 0.6 concentrated is causing the water to be lost from these potatoes. Okay? Are you following? So again, the greater the water, the greater the mass. Same concept, potatoes, the length was 60 and it has decreased. The mass has decreased, okay? Question, did this potato lose or gain water? If its mass or the length, same thing. It has decreased. Do you expect that potato to have lost or gained water? Lost, okay? Water has been moving out, okay? Then the sugar solution has a higher or lower water potential. That solution I have outside here, the blue solution, does it have higher or lower water potential? It must have lower water potential, right? So that the water moved outside. Always water moves from where there's more water to where there is less water. And the potato here must have greater water potential. That's why it lost water. Okay? Question 30. How do concentrations of nitrates and water in plants compare to that of the soil? They said, look what they said. They said that there's a well-watered soil and there is ions being moved by active transports, water moved by osmosis. So, say this is the cell. Do you expect the water to be higher inside or outside the plant root? Here, the water is expected to be higher, right? Why should the water be higher in the soil? Because they said it, look. And what about the ions? They said the concentration of ions inside roots, it should be higher. Why the concentration of ions in the cell should be higher? Because of this word, because they said it has moved by active transport. And active transport is always from lower concentration to higher concentration. So once again, I'm expecting the concentration of the ions inside the cell to be higher because that's the cell and it's active transport here. And I'm expecting the water to be lower because it's osmosis. That's higher water potential, that's lower water potential. Do you get it? Since they said active transport is movement inside the cell, so the cell must have higher. And since they said osmosis, and it's going into the cell, so the cell must have lower water. Okay? Now, for this question, they have removed phloem. Phloem has been removed. Xylem is fine. Nothing's been done to xylem. How is the transport going to be affected? How would you expect the transport to be affected? It's easy, right? Because phloem is gone. If phloem is gone, then water is still fine, right? Salts and mineral ions are still fine. The only thing will be affected is sucrose and amino acids. This is not one of the easiest questions. I want you to take a moment again and try it. Can you read it, then answer? Okay, so the question says, what can you conclude from the graph? Meaning, not everything must come from this graph. Ignore everything you've learned before. The answer should come from this graph. So this is desert plant. This plant lives in the desert. And that is 4 a.m. That's dark, dark, and bright sunlight here in the middle of the day. The question says, here, transpiration shown here in blue. Transpiration is low, right? What else is low during that time? It is also photosynthesis. Photosynthesis is shown here in black. So basically the plant does what? Midday, because it's too hot, like outside. It switches off photosynthesis. When it does this, you know what happens? It closes the stomata. As it closes the stomata, the transpiration is gonna stop. So again, the plant is smart. It's trying not to dry out. It closes the stomata midday when it's very, very hot. Photosynthesis stops, but that has a great benefit. What's the great benefit here? The great benefit, it's not gonna lose water. For that reason, I'm gonna pick B. Most stomata close during light periods. If you don't recognize this, if you don't see this, you don't understand how it works. But that's how it is when it gets too hot. Trying to prevent drying out, close the stomata. You're not going to do photosynthesis, but still, you're not going to die. The plant won't die. Okay? Questions like this are also very important. The questions about limiting factors of photosynthesis. Let me just brief you on how these questions are asked. They give you a graph like this. You have a factor at X. X could be light or CO2 concentration, okay? And the graph is always going to look something like this. Anywhere on the graph where the line is still inclining like this, you have to think of what's on the X. The factor on the X is going to be the limiting factor. Okay? But when I reach that point and the line becomes flat like this, meaning I have enough X. What do I mean here by have enough X? Meaning the value here on the X-axis is too much, right? It's like 10, 20, 50, 60, 100. That means I have enough X. Here, you have to think of other two limiting factors. And if it was light, what are you going to tell them? If it was light on the X, you tell them, there isn't enough CO2. There isn't enough, or there isn't good temperature. Okay? So just think of the other two limiting factors of photosynthesis other than one that they've written for you on X. So I'm going to show you several types of this. X is light. X is light intensity. So at this point here, what could be the limiting factor? I have enough light here, right? It's too bright. So what could be a limiting factor is CO2. How do I know that it's a CO2, not temperature? That's because look up here. This one here has given better result with greater amount of CO2. So always use the best one here at the top. Use it as your reference. Okay? For sure, CO2 is limiting because point 1 gave me better result. Okay? Try another one. Which point with the rate of photosynthesis is limited by light intensity? So where on the graph, where on these graphs, I have light intensity as my limiting factor? First, it's 1. 1 must be one of the answers, right? So it can't be D, it can't be C. Where else light is a limiting factor? It is here, because I have enough CO2, right? There's enough CO2 here, right? So light must be or could be a limiting factor. And even here, temperature is optimum. What prevented this graph from going even higher like this? At this point, because temperature is optimum, it also could be light that is a limiting factor. That's why I'm going for B. Again, find the places where conditions are optimum. If light was in higher amounts, I would have got something like this. And if light was brighter, I would have got the result more like this. And obviously here is dim light, right? It's very early here while it's dim light. Okay? Do you get it? Can you try this yourselves? Okay, so as always, here, it's very bright. I have enough light at this point. So I automatically have to eliminate options that have light as a limiting factor. So get rid of A and B to start with because it's already too bright. And as always, use the best one as your guide, as your reference. The top one is the best one, so compare to it. Temperature here is higher, so temperature gave better result. CO2 is higher, so CO2 also gave a better result. Therefore, it must be option C. Okay? Do you know why we use yeast for like making bread? We know that yeast does anaerobic respiration, but what's the point of using yeast for bread making? It is the CO2, right? Now, option C is correct, but it has nothing to do with bread. Nobody gets drunk if he ate a baguette, right? It's the CO2 that makes the dough rise. Alcohol has nothing to do with it. Actually, alcohol evaporates when you bake the bread. Okay? What happens when left ventricle contracts? So as the left ventricle contracts here, what would you expect to happen to the bicuspid behind? Should close, right? Why should it close? Because it will prevent backflow of blood. So now look at it. The left ventricle is contracting, bicuspid is closing. Meanwhile, semilunar must open. So I'll go for this option. Okay? Super important. And again, you'll find it same drawing. So they don't want students to memorize it anymore. But they change the format here, right? They make ribs higher or moved up, okay? Or the other way around. They just changed the last line, but everything, even the diagram, would look the same thing. Where would you expect the ribs to be? Lowered, meaning the ribs are moving down. So something like this. If his ribs are moving down, what would you expect to happen? You'd expect the pressure to rise, okay? So again, ribs are out, diaphragm is down, the volume will increase, and pressure should decrease, right? So me here, the ribs are pushing on the chest, the ribs are moving down. So what do you expect to happen to the pressure? You squeeze in your chest, you squeeze in the volume of your chest. The volume is going to decrease, therefore the pressure will increase. Okay? Someone's asking me, bicuspid, tricuspid is same as atrioventricular. They mean the same thing. Back to hormones. Remember the correct order? You start with FSH, then estrogen, LH, progesterone. Okay? So once you know them in the right order, full, remember what you used to call them? Once you know the right order, always get it right. Which hormone is not shown? Which hormone is missing? Last question. Tell me which hormone in this diagram is missing. Do you see LH? Yes. Do you see progesterone? Yes. Do you see the very first hormone here, right? The one that starts with, that's FSH, is there, right? It's already there. What is missing? As you can see, the missing one is estrogen. Estrogen rises here. It rises right before ovulation. It should be somewhere here, like this. Okay? Look, if you could just do what I do, literally every paper will take you 30 minutes, 25 minutes. If you start with 2016, 17, you'll start to see like you're scoring 20, 22, 25. By the time you reach 2022, 2023, you'll find your scores getting to 32, 33, 34. You aim for 37. It's very easy paper. It's such a disaster if you score less than 35. Okay? It's really easy. Questions repeat, memorize them, learn them. It's the easiest paper. It's such a good chance to score a good grade here. Okay? All right, I will let you go and I will see you again tomorrow, same time.